So welcome back. Again, we were just talking about chirality and isomers. So again, we have the enantiomers, which are the non-superimposable mirror images. So if you think again about chirality being handedness, so if you think about your hands, they are mirror images of one another, but they are not superimposable. So that's what we're going to take a look at now and in regards to sugars. So we have the D versus the L sugars. Those are enantiomers. So remember, enantiomers differ at every single chiral center. So for example, as I've drawn on the board here, we have D-glucose. So D-glucose, again, has 4 chiral centers. So on these chiral centers, the first chiral center, the third, and the fourth, the OH group is on the right, which means if I were to draw the L version of glucose, this carbon number 2, 4, and 5, normally their OH groups are on the right. I will now draw them on the left because, again, we're looking at a mirror image of one another. So if you notice, all 4 between the D-glucose and the L-glucose, all 4 of those chiral centers have now changed positions where the H's and the OH's are. However, we can take a look at diastereomers. So for example, glucose and galactose differ at this particular carbon 1, 2, 3, 4. So carbon number 4 here, in glucose, the OH is on the right. In galactose, the OH is on the left. Every other chiral center, though, is the same. That's why glucose and galactose here are diastereomers. But you can take the structure of D-galactose now, and you can turn that into L-galactose by switching the position of every single molecule on the chiral center, the H's and the OH, to make, again, those non-superimposable mirror images of one another. The example that your book gives, as you can see in this next slide, right, looks at, again, changing the right or the left of the position of the H or the OH groups to give you enantiomers or diastereomers. So if you notice, D-glucose and L-galactose are diastereomers. D-galactose and L-glucose are also diastereomers because they differ at least one, but not all, stereoisomers. So if you notice, here we have diastereomers, diastereomers, diastereomer, diastereomer, but as we go here to here, left to right, we make enantiomers. So you have many more, right, diastereomers than you do in enantiomers. Now, this sounds very confusing. I'm aware of that. Okay, so I've tried to break it down to make it a little bit easier, but the things to remember are this. Number 1, determine the number of chiral centers. Okay, so let's say that number of chiral centers, we're just going to call it n. Okay, you look at, as you draw your Fisher projection, on the left or the right, do I change my positions of my OH and H groups on either side? If you change at all chiral centers, meaning every single chiral center, the H and the OH change positions. If you change at all chiral centers, you are enantiomers. If you change at 1 to n minus 1, then you are diastereomers. So for example, if you take a look at glucose, glucose has 4 chiral centers. If you change at all 4 chiral centers, you are now an enantiomer. But if you change at 3 or 2 or even 1, you are a diastereomer. So this might not seem like a big deal, but our bodies make a huge difference between these enantiomers. Our bodies only recognize D sugars. And some really cool things, for example, bacteria, what they do in they have what's called a peptidoglycan wall, which is a peptide, which is short for a small little protein. Glycan is what we call just a big bunch of sugars together. At the end of that peptidoglycan, there is a D-alanine. So normally amino acids, we can only recognize L. So if a bacteria puts D in there, our bodies don't recognize it, and therefore we can't kill the bacteria. Bad for us, good for the bacteria. So there are things that you can do that way. For us, we recognize D sugars, but some of these L ones we don't necessarily recognize. So again, it might not seem like a big deal, like you're just flipping these back and forth, that's true, but there are a lot of potential consequences. So for example, if you've ever heard of Parkinson's disease, there is a particular drug that can help alleviate the symptoms for this, and it's called L-DOPA. Not the full name 3,4-dihydroxyphenylalanine, but we'll just call it L-DOPA for short. Well, the L version is actually what helps Parkinson's patients because it's what our brain actually uses in the process for dopamine, which in Parkinson's disease, dopamine levels can be very low, can cause a bunch of symptoms. However, if you were to give a patient with Parkinson's disease D, the enantiomer of L-DOPA, you basically shut down part of their immune system. It lowers their white blood cell count. It doesn't actually help alleviate the symptoms of Parkinson's usually. So again, it might seem very confusing and it might seem like it quote unquote doesn't matter, but our bodies are very, very particular about how things look in 3 dimensions. So again, the biggest takeaway I want you to think about here is that these different stereoisomers can have vastly different properties. So if we take a look, right, now this D versus L, okay, we talked about how we can tell, right, whether or not things are enantiomers or not. However, how do we tell if something is the L enantiomer or the D enantiomer? Well, what you want to do is you want to look and you find the farthest chiral center from the aldehyde or the ketone functional group. So for example, in glucose, here's our aldehyde group. I want to find the farthest away chiral center for this. So this chiral center is the closest. This one right here is the one that's farthest away. So you're looking for that particular group. And so what you want to remember is you want to remember the position of the OH group. If the OH is on the left, that is the L configuration. If the OH, though, is on the right, so I remember L for left, that's the easy way, so then you just remember, oh, if it's on the other side, if it's on the right, then it's D. So for example, let's say I have some random sugar I'm going to make up here just by looking at it. So here's your ketone. And without putting any other piece in, if this OH group here is on the left, this is a chiral center, and it is the furthest one from the ketone here. So this is where you're looking, right above the CH2OH group. This OH group is on the left. This is the L of whatever sugar that is. So if you take a look, when you look at D-glucose, this OH group is on the right. But if you look at the enantiomer with L-glucose, you follow that same chiral center, and look at that. That OH group is on the left. Same thing with D versus L-galactose. The furthest chiral center, OH group here, is on the left. That's the L. The OH group here for D-glucose is on the right. That is the D. So that's how you remember whether or not you're dealing with the L or the D enantiomer. Remember, enantiomers differ at every single chiral center. Okay? So if you take a look, here we have that structure, those structures again, if you look at these slides, of D-glucose, D-ribose, D-glyceraldehyde, etc. Could you draw the L versions of these? So for some practice, I would go ahead and draw the L versions of these and circle which chiral center determines whether you're D or you're L. Could you also draw diastereomers of these? You don't have to name them because that would require you to memorize all the different species, but could you draw potential diastereomers of these particular sugars? So now that we know about monosaccharides, so we know about functional groups, we know about naming them based on the number of carbons, and we know about naming them based on their enantiomer, the furthest chiral center from the highest priority functional group, that glucose or that aldose. Here are some common hexoses, okay, that you may or may not have heard of. So you can see here to the left we have fructose. So fructose is a ketose, but it has 1, 2, 3, 4, 5, 6 carbons. And as I've drawn it here, this last OH group is on the right. So this is D fructose. Here we have glucose. You remember glucose from glycolysis, perhaps. And then we have galactose, which we will talk a little bit more about galactose when we talk about disaccharides. For example, if you've ever heard of lactose before, lactose is a disaccharide, 2 sugars. It's a galactose and a glucose that are physically linked together. So we will see those again. But if you notice, glucose and galactose are both aldoses. But they're all of these are common hexoses, so they all have 6 carbons. So if we take a look really quick, you can do some example problems that can help you. What happens if you could you draw a Fisher projection of a D-pentose? So pentose, think about how many carbons is that? Thinking about the D form, how do we know where we position the OH groups? Okay? Does every single OH group matter, or only a certain OH group matter to make it an L versus a D? And then draw the enantiomer of that. Okay? Then draw a Fisher projection of any aldose sugar you want. But then draw some diastereomer of it. And then finally, draw a sugar that has a ketose group that is also hexose. So think about how many carbons that is. And then draw it as the L enantiomer. And then once you're finished with that, could you draw the D enantiomer? And then could you draw a diastereomer? So getting some practice with this in the homework, or sending me information to see if you have questions about it, please let us know. All right, so now that we know about enantiomers, diastereomers, overall looking at those chemical structures of the monosaccharides, we are now going to go talking about how we put these not from the Fisher projections anymore, but into a ring, because sugars can be present in our bodies both in the linear, what we call the linear form, straight up and down, as well as the ring form. So we will talk about that in our next video.